Oh, where is my mic? 1 second, people. I think so. You are able to hear me and see me. Yes. So today are back with the second lesson of this chapter, which is the structure of DNA, and we will be trying to figure it out what structure of DNA is and what things we have to learn and from where the question will come from this particular topic. Okay. Yes, I am clearly audible and visible. Hi Shri Shai, hi Shivesh, how are you all? So let's start this session. Yesterday, what we discussed? Yesterday, we discussed about the just overview of the DNA discovery that there were 4 scientists. What were the name of those 4 scientists? Hi Bala, how are you? Oh, are you asking हिंदी में yesterday को क्या कहते हैं? Yesterday, कल, कल. Yes. Hi Bala. Okay. So what were the name of the 4 scientists who tried to discover the structure of DNA? Anyone? Anyone in the comment section, let me know. Yes or no? नहीं बताओगे क्या? What were they? They were Altman. Bala, Altman was the person who termed the DNA. He gave the term DNA. We are talking about the structure of DNA. Who gave who gave the structure of DNA? There were 4 scientists. There were 4 scientists. What were they? Watson, Crick, and Wilkins. Yes or no? Watson, Crick, and Wilkins. Clear? Okay. And there were Franklin also. Franklin didn't got the Nobel Prize. This we discussed. Yes? Chalo. Let's talk about the structure of DNA now. The core structure, like how the DNA is made up of, what is the shape, size and all. Let's talk about that today. Is everything fine? Is everything fine? I'm not able to see your comments, guys. 1 second. What happened? Yeah, now it's good. Plant growth and hormone. Yeah. I am planning plant growth and hormone one shot. I'll do it in one shot, Bala. Will that be fine? Okay, chalo. So let's talk about the DNA structure. DNA structure. Okay. So if I talk about the DNA structure, so DNA is composed of what? Polynucleotides chain. Yes or no? What was the polynucleotides? Who remembered the polynucleotides? My PC net is gone. Okay. Chalo. So polynucleotide was what? Polynucleotide means basically lots and lots of nucleotide is present. Respiration. Okay. I'll do it. Don't worry. So here nucleotide was many nucleotides joined together to form what? To form the DNA structure. Just for the revision point of view, I'm going to tell you what was nucleotides. Nucleotides were what, guys? Nucleotides were phosphate. Yes or no? Phosphate plus sugar. What kind of sugar? Pentose sugar. And along with the nitrogen bases. Yes or no? So there is one strand of the nucleotide and there is the another strand of the nucleotide. For example, if this strand starts from point A and ends at point B, so the other strand will what? It will start, yes, it will start from point B and will end at point A. That means both the strand which you are able to see here, is basically what? Is anti-parallel. Is what? Is anti-parallel. Okay, clear to everyone? Yes or no? B2A, exactly, B2A, right? More specifically, if I say that the one strand is basically what? One strand is basically from 3 prime to 5 prime. What is this 3 prime to 5 prime? It's basically showing the phosphate bond, like how the phosphate is arranged. Is it arranged from 3 prime to 5 prime or is it arranged from 5 prime to 3 prime? So if suppose one strand is 3 prime to 5 prime, the other strand will be from 5 prime to 3 prime. Yes or no? Is it clear, Bala, Shri Shai, everyone? Yes or no? Shivesh? Chalo, great. Now what? Do you understand the meaning of complementary? Do you understand the meaning of complementary? What is complementary, guys? What is complementary? I traveled for the in the flight for the very first time, like around 2019 or maybe 2018. I don't remember the year. 2019 not. It was a COVID time. Before that I traveled. So there they use this word complementary, and that complementary word is basically free. So the air hostess came and she handed over me the pack of chocolates, and I was like, they will charge a lot. Like obviously, in planes everything is so pricey. But then that air hostess said that it's complimentary, means it was free. But here that's not the meaning of complimentary, guys. What's the meaning of complimentary is basically, see, we say na, sometimes a group is made. So we say black and white, colored, right? So here what's happening, complementary here what means that there is some groups which is fixed, like A will bond with thymine only. Adenine will make a bond with thymine only. Guanine will make a bond with cytosine only. So here this is the complementary. Okay. Now A and T will make double bond. Yes or no? A and T, exactly. A and T will have double bond, and G and C will have triple bond. Clear? Okay. Great, sir. Any doubts here? Any doubts here? And where these bonds are basically what? These bonds are the hydrogen bonds. These bonds are the, yes, exactly, hydrogen bonds. Hydrogen bonds. What happened to other people? What happened to other people? Bala, Bala and Shivesh, are you there? Are you there? Okay, chalo, great. Now see, now let's move on, and let's talk about, we have seen that there is a rule called Chargaff rule. There is a rule called Chargaff's rule. How many of you have heard about this Chargaff's rule? How many of you have heard about this Chargaff's rule? And from this lots and lots of questions comes. Questions comes from this. What is this? That if suppose there is a double stranded DNA, okay? If there is that double stranded DNA, what is going to happen here, people? What is going to happen in the double stranded DNA? You have heard? Okay, great. So according to this, what happens? That purines binds with pyrimidines, okay? Purines bind with pyrimidines, and number of purines will be equal to the number of pyrimidine. Number of purines will be equal to number of pyrimidines. Number of pyrimidine, okay? So what are purines here? A and G. A plus G will be equal to what? Will be equal to T plus C. That means their ratio should be equal, okay? Their ratio should be equal. So if I write here A plus G upon T plus C will give us 1. So if this is the case, the DNA is what? The DNA is double stranded DNA. The DNA is what? Double stranded DNA. Clear to everyone? If this is the case, the DNA is double stranded DNA. Should I give you a question? Should I give you a question related to this? One second. Identify the double stranded DNA, okay? Identify the double stranded DNA. What you have to do is that you have to identify the double stranded DNA. Now how you are going to identify? I'm going to give you the question. So here I am saying that I'll give you this option. One second. I'll make here we have purines and here we have pyrimidines, okay? Pyrimidines. So I am saying that in option A, purines are 90% and pyrimidines are 10%. In option B, I am saying that purines are 60% and pyrimidines are 40%, right? And then I am saying that purines are 50% and pyrimidines are also 50%. And in D, I am saying that purines are 0% and pyrimidines are 100%. What could be your answer? What could be your answer? Option C. Everyone, option C. Is that so? Everyone option C. Shivesh, Bala, everyone option C. Yes, obviously. Why this is option C, guys? Why this is option C? Because right now we said that how to identify the double-stranded DNA. If we need to identify the double-stranded DNA, Chargaff said what? Chargaff said that ratio of purines and pyrimidines should be equal to 1. And if it is there, then that DNA is considered as double-stranded DNA. Clear to everyone? Any doubts anywhere? Any doubts anywhere? No? Great. Now, next we are going to go with the base ratio. What is base ratio? What is base ratio of our DNA? See, don't get confused with the Chargaff rule and the base ratio. Chargaff rule is different. Base ratio is different. Base ratio we calculate as adenine plus thymine upon G plus C, guanine plus cytosine. Base ratio we do adenine plus thymine and guanine plus cytosine. Clear? Okay, everyone? Yes or no? Great. Just give me a second. Just a second. Oh, 1 second. Okay, chalo, great. So base ratio what we have here? A plus T divided by G plus C. Okay. Now, this ratio is constant for a species. This ratio is what? Always constant for a species. For a species. Let's suppose that for human, I'll say this ratio is 23. Just a random number. So for all humans, whether you Uma Shankar sir is basically traveling. He is traveling. He will resume the classes very soon. Don't worry. Sir is traveling basically. That's why. Okay. He will continue the classes, Shrishan. Don't worry. Chalo. So base ratio is constant for a species. Clear? Okay. Now, next see here. If suppose that 80 ratio is more. If I'll say that 80 ratio is more than G plus C, so this type of DNA is known as what? 80 type of DNA. This type of DNA is known as 80 type of DNA. And similarly, guys, if suppose GC ratio is more, if GC is more, if G plus C is more, then what is going to happen? The DNA will be what? The DNA will be GC type of DNA. Okay, the DNA will be GC type of DNA. Clear? It's a very easy. Yes or no? It's easy. Yes or no? Yes. Chalo. Now, acha. If I am talking about the 80 type of DNA, so this type of DNA is basically what? This type of DNA are the eukaryotic DNA. They are what? They are the eukaryotic DNA. And GC rich DNA is basically what? It is prokaryotic DNA. Okay, prokaryotic DNA. Clear? So we can say that eukaryotic DNA are those type of DNA which is 80 rich, and prokaryotic DNA is basically the GC rich. Clear to everyone? Any doubts? Any doubts, guys? Any doubts? No doubts? Great. Chalo. Now, do you remember, like, if I ask you, if I give you 2 DNA, okay, and if I ask you which DNA, okay, I'll give you the question. Okay, our DNA is made up of, our DNA is made up of 70% of no doubts, great. Our DNA is made up of 70% of A plus T base pair, and another DNA has 30% of, okay, not 30%, let's take another DNA has 60% of AT base pair. Which DNA will have 50% more melting point, or which DNA will have higher melting point? Higher melting point. Tell me the answer. A very good question this is. Which DNA will have the higher melting point? So what is here? We have two types of DNA, right? One DNA is basically what? One DNA is having A plus T and the 8 plus T as 70%, and the other DNA is having A plus T as 60%. So which DNA is going to have more higher melting point? Is it A? Is it A or B? Okay, Sri Shai said A, Shivesh said B. Okay, okay, any other one? Any other person who wants to answer this question? So guys, if I talk about this question, so DNA B will be having the higher melting point. The one which is GC rich, the one which is GC rich, is going to have higher melting point. Okay, clear to everyone? The DNA which is GC rich is considered to be as the higher, having the higher melting point. Clear? There can be one reason. I think first one will have more organelles. Organelles? Okay. No. So GC will have what? GC. See, not 3 hydrogen bond is there. So we have to give more and more energy to break these bonds. Yeah, right. So automatically the melting point will be increased. Yes or no? Yes. Great. Chalo. Now let's move on and let's talk about some other facts about DNA. I'll try to start that search for this genetic material topic also today. Yes or no? Chemical structure. Yes. Chalo. Now let's talk about the configuration of DNA now. Configuration of DNA. So we talked about this thing, right? Ki DNA is made up of what? DNA is made up of phosphate and sugar and nitrogen base. So I am not writing it again, okay? And we have also discussed that it is having polarity, like it's made up of 2 chains. Suppose that this is the one chain of DNA. I'll make this DNA here. This is the one chain and this is the another strand of DNA. So one strand will be what? It will start from 5 prime and it will end to 3 prime, and the other strand will start opposite. Yes or no? So it will start 5 prime here and 3 prime here. Clear? This is to no doubt anyone. And this DNA is made up of what? Phosphate. This white color molecule is the phosphate, and this one is the what? This one is the nitrogen base. Let me draw here. This one is the nitrogen base. Again, this white color is the phosphate. So phosphate and sugar is what? Phosphate and sugar is the backbone, while nitrogen bases are acting like the stairs. Yes or no? Yes. Great. Any doubts? No. Okay. Now, what is the distance between 2 successive nitrogen base? See, one nitrogen base is here, one nitrogen base is here. इन दोनों के बीच में what's the difference? What is the distance between 2 successive nitrogen base? So guys, you have to remember this. The distance is basically 3.4 angstrom. Yes, 3.4 angstrom or 0.34 nanometer. 0.34 nanometer. Clear? The distance between these two nitrogen base pair, bonds of nitrogen base pair, is basically what? 3.4 angstrom or 0.34 nanometer. Okay, clear? Now in one turn we are having 10 bonds. In one turn we are having 10 bonds. What is one turn? See here, this is the one turn, this is the one turn. So in one turn what we are having? In one turn we are having 10 nitrogen base. So what will be the length of one turn? What will be the length of one turn, people? Tell me, what will be the length of one turn? अगर एक टर्न में 10 है, if one turn is having 10 nitrogen bases and each nitrogen bases is having, yes, exactly, the distance of each nitrogen base is 0.34 nanometer. So 10 will be having what? 0.34 multiplied by 10, yes or no? So it will be 3.4 nanometer, or I can say 34 angstrom, yes or no? So this length is basically what? This length is basically known as the pitch of DNA or the helix length, okay? Or the helix length or pitch of DNA. Clear to everyone? Any doubts? Any doubts? Yes or no? Any doubts? Now, what's the distance between these two? What is the distance between the two strands of DNA? What is the distance between the two strands of DNA, guys? It is 20 angstrom. It is 20 angstrom. Okay? 20 angstrom. Clear to everyone? Distance between two strands of DNA, 20 angstrom. Distance between one helix, length of one helix is 34 angstrom, or it is 0.34 angstrom, the distance between the two consecutive nitrogen bases. Clear? Any doubts? Great. Great. Now what? Now we have what is the, like, see, the DNA is kind of helical model, right? So that means what? If one nitrogen base is here, the second nitrogen base is little bit forward, then the third nitrogen base is little bit more forward, like this, right? So they are having what? They are having some variations of degree also. They are not arranged like this. They are arranged like this. Yes or no? Yes. So the variation of degree is basically what? It is 36 degree. 36 degree. Yes or no? 36 degree. So, yes, I'll write here. Each nitrogen base, each nitrogen base pair have the variation of 36 degree, have the variation of 36 degree. Yes or no? Clear to everyone? Any doubts anywhere? This is right. 10 स्टेप के बाद दैट्स फाइन है, वो 360 डिग्री टर्न ले रहा है. सोचो ना, खुद सोचो कि अभी क्या था, यहां से यहां तक एक टर्न लिया, फिर दूसरा टर्न लिया, तो क्या हो रहा है, जब हेलिक्स बना रहा है तो 360 डिग्री 10 बेस पेयर आने पे 360 डिग्री कंप्लीट हो जा रहा है, तो एक टर्न हो गया. यस और नो? यस और नो? बोलो. क्लियर? Great. चलो, I'll move forward. Someone is saying, someone is not. शिवेश is saying, हाँ ये तो सोचा ही नहीं था. अब सोच लो शिवेश, right? अब सोच लो. चलो. Great. Now let's have some. We have to learn some of the data of different types of. अब समझ में आ गया क्या? अब समझ में आ गया क्या? अब तो नहीं रट्टा मारना पड़ेगा. चलो, बट कुछ चीजें हैं जो तुम्हें रट्टा तो अभी भी मारनी पड़ेंगी, जैसे जो मैं अभी लिखने जा रहा हूं. 5174 बैक्टीरियोफेज. क्या है? 5174 बैक्टीरियोफेज. बैक्टीरियोफेज. I'll ask Uma Shankar sir that we were missing him, right? So 5 bacteriophage is basically contains how much type of nucleotides? 5386. How much? 5386. Yes or no? Now we have bacteriophage lambda. We have bacteriophage. Yes. ये याद था क्या? Bacteriophage lambda. How much? How much do we have? How much do we have? 48502. 48502. Remember this, I talked about the nucleotides. This I talked about the nucleotides. Galat? Okay. And this is what? Base pair. I am talking about the base pair. Basically, base pair. Okay. Now, next what do we have? E. coli. We have to remember. We have to remember of E. coli. जो हमारे जिंदगी में तबाही मचा के रखा है. Yes or no? नहीं भाई, E. coli is a good bacteria. E. coli is a good bacteria. So what about E. coli, guys? E. coli, if I talk about, so it is having 4.6 into 10 to the power 6 base pair. Yes, very good. 4.6 into 10 to the power 6 base pair. Okay. And next, what do we have? We have human cell, which we have to remember. ये तो तुम्हारा अपन है, इसको तो याद रख लो. Human cell. I'm talking about the haploid content. Okay. So this will be what? 3.3 into 10 ka power 9 base pair into 10 ka power 9 base pair. Exactly. 9 base pair. Yes or no, everyone? Yes or no, everyone? Great. Great. Chalo. Now, this much done. This much done. Now we will be talking about the diploid is 6.6 into 10 ka power 9. Yes. Now we will be talking about the types of DNA. Now we will be talking about the types of DNA. So, see, this starting portion of this chapter is very much rectification wala thing. Neither I can do in this thing anything, nor you can do. You have to learn these data. And when the concept will come, so we will understand the concept. Right now, I am giving you the data, what things you have to remember. See, in some of the books, lots and lots of things are there. We don't have to remember so many things. We don't have to become a scientist, right? So whatever needed in NEET, I am covering it. Okay, chalo. Now, let's have types of DNA. Types of DNA. What's the meaning of bacteriophage? Bacteriophage is the type of organism. Okay, it's a name of an organism. Types of DNA. Types of DNA. Okay, now see here. So first of all, we have right-handed DNA. We have right-handed DNA. What handed? Right-handed DNA. That means what? Clockwise twisting. ऐसे तुम ऐसे इमेजिन करो कि तुमने दो चीज लिया. क्या लिया? You have taken two threads. Okay, and you have hold those two threads and उन दोनों को तुमने clockwise twist कर दिया. क्या किया? Clockwise twist कर दिया. So that type of DNA is basically what? That type of DNA is known as right-handed DNA. Right-handed DNA. It's also known as BDNA. It's also known as BDNA, and this was the only DNA on which this Watson and Crick model proposed. This DNA was proposed by Watson and Crick. Watson and Crick का जो मॉडल था वो BDNA या राइट हैंडेड DNA को ही शो कर रहा था. Is it clear to everyone? हाँ जी बताओ बताओ. Is it clear to everyone? Great. चलो. Now, if I talk about this BDNA, so normal DNA is basically the BDNA. Whatever the structure we have read is. ये क्या हो गया? 1 second, guys. ये बताओ, ये यहीं रह गया और ये निकल गया. हाँ? 1 second. Hi. Your name is so. What should I pronounce? Chalo. Now, if I talk about the B type of DNA, so normal DNA which we are studying, that is only the B type of DNA. So we have what? We have 34 angstrom helical length. The helical length of BDNA is 34 angstrom, which we have read. 10 pairs in one turn. Yes or no? 10 pairs in one turn. And हर एक नाइट्रोजन बेस के बीच में कितना डिस्टेंस? 3.4 डिस्टेंस. यस और नो? यस और नो? ओके. एंड डायमीटर कितना? 20 एंगस्ट्रॉम. सो जो डीएनए के बारे में, जो नॉर्मल डीएनए के बारे में हमने पढ़ा, वो बेसिकली क्या है? बीडीएनए. क्या है? बीडीएनए. चलो, ग्रेट. अब देखो यहाँ पे लेफ्ट हैंडेड डीएनए क्या आ जाएगा यहाँ पे? लेफ्ट हैंडेड डीएनए. दूसरा जो है लेफ्ट हैंडेड डीएनए. व्हाट इज दिस लेफ्ट हैंडेड डीएनए सर? सो बेसिकली left-handed DNA is what? You have taken two strands of DNA. दो strands of DNA तुमने लिया और उसको anti-clockwise twist कर दिया, तो वो क्या हो गया? Left-handed DNA. What is it? Left-handed DNA. So anti-clockwise twisting. Anti-clockwise twisting is present. So we will be going to call it as left-handed DNA. She wants you to, sir, to call her by his. Yeah, anti-clockwise twisting. Anti-clockwise twisting. Yes or no? And example of such type of DNA will be what? Example of such type of DNA will be, then you guys will make meme on me, right? Example of such type of DNA will be what? Um, ZDNA. It is ZDNA. Okay? ZDNA. Clear? So this thing you have to remember. Ab left-handed DNA में what you have to remember is that number of base pair. Just remember the number of base pair. So number of base pair in one helix. Number of base pair in one helix is what? One helix is 12. One helix is 12. Okay? हमारे में कितना है 10, इसमें कितना है 12, इसमें कितना है 12. समझ में आ रहा है कि शिवेश, अक्षय, जे, एवरीवन? बाला बाला तो चली गई लगता है. यस और नो? ओके, चलो. नाउ लेट्स मूव ऑन एंड लेट्स नाउ टॉक अबाउट दी packaging of DNA. So you might be very much confused about some terminologies: chromatin, chromosomes, nucleosomes, solenoid, right? So all the things I will be clearing it, okay, today. So see here, packaging of DNA, how the DNA is packed in your chotu sa cell. packaging, packaging of DNA. Packaging of DNA, okay? Yes or no? Yes or no? Okay. Chalo. What, Shivesh, what you are saying? Acha. Sir, you live in? I live in a house. I have a house. I am from Ranchi, Jharkhand. I am from Ranchi, Jharkhand. Not leave. Yeah, exactly, Srisha. That's what I was thinking, like what he is asking. Okay, chalo. Should I ask some English teacher also to take classes? Don't worry. I always say my students that if you are, it's just a thing, like if you are good in English, your biology will be good because you read NCERT in English language, right? But it's not like that. Hindi medium students also learn biology very well. Okay, let's get back here. Packaging of DNA. Okay, packaging of DNA. So see here, there are two types of cell. We have prokaryotes. We have, where is Ranchi? We have prokaryotes and then we have eukaryotes. Okay, it's in Europe, Akshay. It is in Europe. One is prokaryotes and the other is eukaryotes. Okay? So prokaryotes are what? You remember prokaryotes were having the old DNA and they were not having the nuclear membrane, yes or no? So in prokaryotes, DNA is found where? In prokaryotes, DNA, it is found in the cytoplasm only. It is found in the cytoplasm only, yes or no? It is found in the cytoplasm only. पढ़ लो अक्षय थोड़ा, है ना? पढ़ लो. Yes? Chalo. See here. So we have DNA and we have the cytoplasm. We have DNA and we have the cytoplasm. And DNA, where it is present? Present in the cytoplasm of prokaryote. How it is present? Supercoiled. How it is present? Supercoiled structure. In a form of supercoiled. But if I talk about nucleoid, yes, exactly. Supercoiled in India. No, it's in Europe. Guys, it's in Europe. What, Srisai? It is in Europe. Okay, chalo. So DNA is present in the cytoplasm and it is present in the form of a supercoiled structure. And this coiling is done by the non-histone proteins. In prokaryotes, there is no histone proteins. So the coiling is done by non-histone proteins. This point you have to remember. This is what you have to remember, that in prokaryotes, coiling is done or packaging is done by non-histone proteins, okay? Non-histone proteins. And the supercoiled structure is now known as what? Nucleoid. What's the another name for large loops? Yes. What's the another name for nucleoid? What's the another name for nucleoid, guys? Akshay, Srishai, Shivesh, another name for nucleoid. Another name for nucleoid. Another name for nucleoid. Shivesh is saying Ranchi is in Mars. Okay. Yes, very good. It's what? It's what? You think what, Srishai? What do you think? That Ranchi is in Mars? Or do you think that the another name for nucleoid is genopore? What is it? Genopore. Okay? Genopore. What's another name for nucleoid? It's genopore. Clear? Okay. Now let's come back to the eukaryotes, and I'll write eukaryotes in the next page because आधा जगह तो मैं ही ले रहा हूँ. So I'll write eukaryote in next page. Eukaryotes. You didn't know? Okay. Note it down then. Note it down. So now what's the eukaryote? So in eukaryotes we have what? We have lots and lots of things. Eukaryote का packaging हमें बहुत अच्छे से देखना है. See here. First of all, I'll make a diagram and then we will understand. So we have what? We have this DNA. Okay. This is what? DNA. पहचानो इसको, तुम्हारा DNA है ये. Okay. Now, this DNA is going to do what? This DNA will going to attach with the histone proteins. This DNA is going to attach with the histone proteins. Yes or no? Okay. Histone proteins. Now guys, if I talk about the histone proteins, it is of 5 types. Yes, it is of 5 types. H1, H2, H3, H4. Yes or no? And H5 is also there or what? Is it so? H5 is also there or what? Is it so? No guys, it's H2A, H2B, H3 and H4. Yes or no? And there is one which is known as H1. So this is the name of the proteins. This is the name of I'll write it everything. Don't worry. I'll write it everything. Okay. So first of all, let's see here. Histone proteins are there. Let's understand. Let us imagine it. Okay. So suppose that this is the charger. ये चार्जर का तार है. Okay. एंड यस H2A H2B ये चार्जर का तार है और ये हिस्टोन प्रोटीन है. ओके तो डीएनए क्या होगा, इसके ऊपर ऐसे करके लपटा जाएगा. ओके क्लियर? यस और नो? डीएनए विल बी coiled around this histone protein. Yes or no? Okay. So the DNA structure will be looking like this. The structure will be looking like this. Let's suppose that these are the histone proteins. Yes, histone octamer. These are the histone proteins. We'll talk about the histone octamer thoda der baad. Okay. These are the histone proteins and DNA is like they are so coiled here. They are what? They are coiled like this. Okay. They are coiled like this. Yes or no? So this structure is known as what? This structure is known as nucleosome. This structure is known as nucleosome. And when you will see, it's looking like what? It's looking like that you have that, you know, necklace and moti laga hua, pearl laga hua hai. So we say that beaded structure. This is known as what? Beaded structure. Yes or no? Beaded structure. It looks like the beaded structure. So what is nucleosome, guys? Nucleosome is nothing but the DNA plus histone protein. Yes or no? Nucleosome is what? Nucleosome is the beads on string structure. Yes, beaded structure. Nucleosome is what? Nucleosome is the on. Yes, nucleosome is what? It is the what I was saying. Nucleosome is what? Nucleosome is the DNA plus histone protein. Okay? Yes or no? Clear. Great. Chalo. Now, after that what? After that there will be presence of some non-histone protein. Then what will come? Some non-histone protein will come. Non-histone complex will come. And then again there will be coiling. And now what it will form what? It will form solenoid. It will form what? It will form solenoid. Okay. Now it will form solenoid. Solenoid. So alone histone protein is not able to condense the DNA so much. So it takes the help of non-histone complex and then non-histone, with the help of non-histone complex, this nucleosome gets converted into solenoid. and I have written the spelling of solenoid so wrong. Solenoid. Zoned out. Yeah, sometimes it happens. Solenoid. Okay. So what we saw? DNA plus histone protein, nucleosome. Nucleosome plus non-histone protein, solenoid. Yes or no? Clear? Okay. Then the solenoid converts into super solenoid. This solenoid converts into super solenoid, more intense coiling. More intense coiling. This is known as what? This is known as super solenoid. I think so. Hi, midlife. How are you? I think so. Stopped for a while now. So Shivesh is saying that only. So this is what this is, super solenoid. This is super solenoid. Yes, Shivesh? Super solenoid, right? And then after this super solenoid, then we have what? Then we have the chromatids. Then we have the chromatids, okay? Then we have this chromatids, and finally the chromatids gets converted into the supercoiled chromosome. Supercoiled chromosome. Okay, everyone, see here. See here. Is it understandable? Is my diagram understandable? So how it's happening? It's happening first. We have the normal DNA. Then this DNA gets what? This DNA gets histone proteins, yes or no? And then histone proteins, with the help of the histone proteins, we get nucleosome. Nucleosome. Now nucleosome के पास फिर से कॉम्प्लेक्स आता है, नॉन हिस्टोन कॉम्प्लेक्स. Non histone complex, and it makes what? It makes the solenoids, and then from the solenoids we have what? We have super solenoid. Toroid यहाँ नहीं आएगा, it's of your physics part, right? बाप रे, I remember that solenoid toroid बहुत परेशान किया मुझे इसने 12th में. Super solenoid. After this what will happen? Then we will have the chromatids, and then we have what? We have the chromosomes. Then we have the chromosome. Clear? Chromosome. Any doubts? Any doubts? Chromosome. Yes or no? Shrishai, if it would be easy for me, then I would be a physics teacher right now, right? For me, physics was out of the box. Yes or no? Okay. Let me know. Is this clear? Is that clear? Is this clear? Chalo, should I move forward? Akshay, are you listening? Akshay, is it clear? Next, what do we have? Whom do we have? Midlife, is it clear? Your physics is epic. My physics is very epic, Shivesh. Okay, chalo. Now, let us talk about some of the things. थोड़ा हिस्टोन ऑक्टामर और ये सब के बारे में पढ़ लूँ, बता दूँ थोड़ा. See, first of all, what's the basic difference between the packaging of DNA of in prokaryote and eukaryote? So in prokaryote, they only use non-histone proteins, and in eukaryotes we are using what? We are using the histone proteins. Now histone proteins are rich, and I'll give you the question. Histone proteins, with the help of question, let's remember, histone proteins is rich in number one is valine, number two is arginine, number three let's have lysine. Acha, you heard that I was taking physics class. Lysine. And second is both B and C. Both B and C. So what will be the answer here? What will be the answer here? What will be the answer here? Both A and C. Huh? Both A and C. Shrishai, both A and C. Both B and C, na? Both B and C, exactly. So, yes, not A and C. Both B and C. Valine is not the answer. Arginine and lysine. So this histone protein is what? This histone protein is rich in two amino acid, which is arginine and lysine. Okay, this is a very favorite question for NEET exam. Like they sometimes ask this. So our DNA is made up of what? Our DNA is, our histone protein is made up of what? Histone protein, which we use for the packaging, is rich in, histone is what? It is rich in these two amino acids, which is what? Which is lysine and arginine. Lysine and arginine. Clear? Lysine and arginine. Okay, we will complete this, guys. Okay, we will complete this thing, then we will go. From tomorrow we will start the search for genetic material. So thoda sa late hoga, but we will complete it. Okay, histone is rich in lysine and arginine. Clear? And it is what? Is it a basic protein, acidic protein? It is a basic protein. It is what? It is a basic protein. Basic protein. It's not acidic. It's not acidic. It's a basic protein. Yes or no? Clear? Great. Now, 5 types of histone are there. How many types? 5 types of histone. What we have? H1 histone. Then we have what? Then we have H2A. Then we have what? Then we have H2B. And then we have H3. क्या क्या है? Positively charged. Yes. H1, we have H1 histone, H2A, H2B, and then we have H3. How many types? How many types? 5 types. Okay. अच्छा, एक चीज तो मैंने बोल गया, H4 and H4. Yes or no? We have this type of proteins. कितने proteins हैं हमारे पास? 5. अब देखो, ये ना H1 जो है, H1 क्या है? Not लगाने का काम करता है. What does H1 do? Just remember that H1 is that kind of protein which makes a knot. H1 makes what? H1 makes a knot. How, sir? See, we took this example, right? This was the histone protein. इसको ऐसे लगा के अगर छोड़ दोगे तो ये क्या होगा? खुल जाएगा. तो H1 क्या करता है? H1 आके यहाँ बैठ जाता है. H1 क्या करता है? H1 यहाँ पर आके बैठ जाता है ताकि ये नॉट जो है वो खुले ना. क्लियर? क्लियर? बाकी H2A, H2B, H3 एंड H4 क्या बनाते हैं? ऑक्टामर बनाते हैं. कुछ इस तरह से अरेंज्ड हो जाते हैं. दे आर अरेंज्ड लाइक दिस. 4 आगे 4 पीछे, 4 आगे 4 पीछे. क्लियर? इन दिस वे दे आर अरेंज्ड. सो इसको क्या बोलते हैं? यस, एग्जैक्टली. इसको क्या बोलते हैं? ऑक्टामर. इसको क्या बोलते हैं? सॉरी, दिस इज एन इंग्लिश चैनल एंड I am speaking in Hindi. I'm very sorry. So this is known as what? This is the octamer. Guys, you all, whenever I start speaking in Hindi, na, you guys comment, sir, English, okay? Chalo. So this is known as the octamer. And this is the DNA. And outside this complex, H1 is present, right? So H1 is what? H1 is not the part of octamer. Okay, I'll make the diagram again. See here. This is the histone octamer. This is the histone octamer. Okay, a beautiful diagram I'll try to make. And this is our DNA. This is our DNA. It's like this. It's like this. Okay. And yes. Now, this is what? This is histone octamer. Octamer histone. Except H1. Every protein is present. Now, H1 will be where? H1 will be here. H1 will be here. Okay. Right? H1 will be here. Clear to everyone? Any doubts? Any doubts? Yes. But Sri Sai, this is an English channel and my team will be like, sir, why you are not teaching in English? Then what? Then what I'll do? Huh? Okay. Chalo. Great. Clear? Clear? Any doubts? Any doubts? Chalo. If not, let's move on. Let's move on. Great. One second. One second, guys. Yes. Now see, if I talk about, suppose this is what? This is one histone octamer. This is the another histone octamer. This is the another histone octamer. And the DNA is present here, right? Like this. Like this. So see, there are some DNA which is present between the two nucleosomes. There are some DNA which is present between the two nucleosomes. So such type of DNA is known as what? They are known as linker DNA. It is known as what? Linker DNA. Yes or no? what we will say linker DNA. इस DNA को क्या बोलेंगे? Linker DNA. Any doubts? Any doubts? No doubts? Great. Great. Okay. Now see, if I'm talking about the non-histone proteins or non-histone complexes, if I'm talking about the non-histone complexes, is it acidic or basic? Is it acidic or basic? Non-histone proteins acidic होंगे या basic होंगे guys? Tell me. Non-histone complex will be acidic or basic? Histone तो it will be basic. It's for sure. No. Remember that non-histone proteins are what? Acidic. Non-histone complex are what? Acidic complex. Okay. This is acidic. Okay. Histone is basic, but non-acidic protein. Why I'm saying non-acidic? Non-histone complex is acidic. Okay. That means to make solenoid, we are using the non-histone proteins. So those non-histone proteins are what? They are acidic in nature. Clear? भूलोगे तो नहीं? Okay, you learn it. Just have a thing in your mind. Okay. Now, see, in chromosomes, what happens? Let's suppose that this is the chromosome. Let's suppose that this is the chromatin. I will say. I'll suggest chromatin. Okay. So see here. This is one chromatin. And we have another chromatin. Then we have this chromatin. Then again we have this chromatin. So can you see there are some places like here and here. They're what? They're very dense. They are what? They are very dense. When you color it, when you apply stains, they will look like a very dark stained structure. Dark stain. They will look like a very dark stain. Yes or no? Dark stain. Now such type of area is known as what? Such type of area is known as what? Heterochromatin. Such type of area is known as heterochromatin. Heterochromatin. Okay. Heterochromatin. This is what? This is the heterochromatin. Okay. Now if I talk about this heterochromatin, guys, they are transcriptally inactive. They don't do anything. They are what? They are inactive. They won't make RNA. They won't make protein. So they are what? They are inactive. Okay. Inactive. Yes. And these are what? These are the light structure. These are light structure loosely packed. So these are known as euchromatin. These are known as euchromatin. And these are the structure which is transcriptally active. They are transcriptally active. That means they will make the RNA, they will make the protein. Okay. Clear to everyone? Any doubts? Euchromatin, exactly. Euchromatin, true chromosomes. True chromatin also. Heterochromatin is basically what? Dark stain. Yes or no? Clear? Any doubts anywhere in the structure of DNA? Any doubts anywhere in the structure of DNA? Everyone let me know in the comment section. Everyone let me know in the comment section. Tomorrow what we are going to do? Tomorrow will be a very special class for you all guys, because we will be completing the search of genetic material. Okay, we will be completing the search of genetic material and I'll bring some questions. जहाँ तक हमने आज पढ़ाई की है उसका मैं कुछ questions लेकर आऊँगा. We will practice some questions. Okay, yes, Shri Shai, please ask me. We will practice some questions and then we will start the search for genetic material. Okay, yes or no? कल सब माइक्रोस्कोप लेकर आना, हम ऐसे ढूंढेंगे जेनेटिक मटेरियल को. ओके, टेल मी अबाउट द डाउट्स श्री शाइन. टेल मी अबाउट द डाउट्स. शिवेश, डू यू हैव एनी डाउट्स? डू यू हैव एनी डाउट्स? Yes, any doubts? I am not able to see your comments. So does H1 take part in nucleosome formation? No. Nucleosome formation, yes, it takes. Nucleosome formation it takes. Octamer का part नहीं है ये. H1 is not the part of octamer. H1 is not the part of octamer, not the part of octamer. Okay, but the whole structure is known as what? Nucleosome. This whole structure is known as nucleosome, right? So nucleosome, it makes nucleosomes but not the octamer. Okay, okay people, clear? Okay सर डीएनए रेप्लिकेशन का क्वेश्चन लाना. ओके ओके फर्स्ट पहले पढ़ ले डीएनए रेप्लिकेशन, फिर तो क्वेश्चन लाऊंगा ना मैं शिवेश. हाँ okay, I will definitely, I'll definitely. Guys, I am doing one thing in the Unacademy plus, not plus, sorry, not plus, in Unacademy free platform. From 5 to 6 I am taking question series. Okay, tomorrow most probably we will do the PYQs of genetics, all the PYQs of genetics. So if you guys have time and if you want to solve the questions, you can come and attend that session. Okay, chalo, bye bye everyone, see you in the next class. And tomorrow the class will be on YouTube. It will be 6:30 to 7:30 as usual, no changes. Okay, chalo, bye Shivesh, bye Shri Shai, bye Akshay, and whoever is here, bye Midlife if you are watching. Chalo, bye bye everyone, see you in the next class.